It actually started with an observation in mice. That is a very old observation that was made in my group. We found that STAT1 deficient mice, these are mice that are lacking a particular transcription factor, STAT1, develop a myeloid proliferative disease. And interestingly, if we then transplant this disease or treat the mice with drugs that suppress the myeloid disease, these animals subsequently developed a very aggressive B-cell disease that manifested itself in the bone marrow, in the spleen, and also in the lymph nodes. This is unusual, and at the same time, Ulrich Jäger and Heinz Gieslinger and their team at the Vienna University Clinic observed that patients that they treated, MPN patients that they treated with ruxolitinib, which is a JAK1/2 inhibitor that would also block STAT1, and that is the common ground for our observations, that some of these patients also developed a very aggressive B-cell disease that looked like the one that we observed in our mice. And that brought us together. We are coming from the mice, having this myeloid disease that switches into a B-cell disease, and the clinicians that also had the myeloid disease treated their patients with JAK inhibitors and also ended up in a certain subset of these patients with a B-cell disease. And I think the major breakthrough is that we not only found this connex, but that Ulrich Jäger and Heinz Gieslinger were also capable to identify the patients at risk. They found that patients that have not a malignant, but patients that have a B-cell clone, that already at the time point of myeloproliferative disease have a hidden B-cell clone that is amplified, that these patients are actually at risk to develop the B-cell malignancy. So what we would propose is that any patient that receives JAK1/2 inhibitors or ruxolitinib should be pre-screened for the existence of this B-cell clone, and this patient should be excluded for safety reasons. And the same B-cell clone was found in our mice, so also our mice have the B-cell clone present already when they manifest themselves with the myeloid disease. That then comes through if we take the myeloid cells away. And we think this is very interesting and exciting, and it's probably the B-cell clone, the taking away of the myeloid cells, and the tumor and the immune suppressive effect that all comes together to make this novel disease. And what was also interesting, we did RNA-seq analysis of the mice, of the murine tumors and the human tumors, and we found that they are resembling each other. So we have probably a disease entity here that really has common grounds, and the mouse could be very valuable to study this disease in more depth. Our findings in Vienna were confirmed in France. So there's a French cohort of patients that were also treated with ruxolitinib, with JAK1/2 inhibitors, and the result that patients with a B-cell clone manifest themselves with a malignant B-cell disease, they confirmed our data. So there's not only the data from Vienna, but also data from Paris and from France.